Hello everyone, welcome to this Protein Simple webinar entitled Your Protein Sample Deserves the Best Detection. Wes gives you 100 times more dynamic range at 3 microliter sample volume. My name is Patricia Piatti, the Simple Western Product Manager, and I will be the moderator. We have very exciting news to tell you, so let's get right to it. Our Senior Vice President, Bob Gavin, will start today's presentation by introducing our company and Simple Western. After that, Dr. Chris Hager, manager of our Application Science Group, will present the new features that make Wes able to give you a broader dynamic range with 40% less sample. And with that, I will hand over the presentation to Bob.
Thank you, Patricia. We're going to talk today about your sample. Most of the time, the most important thing, the most valuable thing in your lab is the sample that you've been working on. That sample might be a tissue sample from a patient. It could be a complex cell lysate. It could be a protein therapeutic or anything in between. But that's a sample that you put weeks, days, hours, long time into developing and making. And you've built that sample in order to answer some kind of scientific question. We focus on trying to make that path between the scientific question that you're trying to ask with your sample and the answer that you're trying to derive as simple and easy as possible. Up till now, many of those processes that you apply to answer that question are complex and convoluted. What we provide are systems to allow scientists to focus on their scientific questions and their answers, not the difficult experiment that they run to get between those two. In order to do that, we apply a deep technical competence across a number of fields, be that imaging or separation, as well as immunoassay and microfluidics. And we apply these in systems that do measurement and analysis of protein solutions of all types to allow scientists to characterize and measure their proteins in new and innovative ways. We do this using 6 different product lines. Those 6 product lines being our imaging product lines, the Fluorochem and the Alpha Imager, the Simple Western, which we'll talk about today, the single cell product line and the Simpleplex product line, as well as ICE and MFI. The first 4 of those product lines are focused primarily on complex samples where you're trying to detect and measure a single protein in a complex mixture of many proteins, something like a cell lysate or a tissue sample. These can also be used on proteins that are purified, simpler protein solutions. The ICE and the MFI products, on the other hand, really focus on these. They're used primarily for protein therapeutics, proteins used as drugs, and are used throughout the entire process of therapeutic development, as well as QC. We have a proven track record of innovation, continually introducing new systems, new versions of our systems, as well as modifications and improvements in our systems over a number of years, always focused on trying to provide you with the most efficient and simplest way to do the scientific research that you're trying to perform. Protein Simple, the Protein Simple brand, is part of a family of brands offered by Bio-Techne. Bio-Techne being the parent company of a number of brands, those including Protein Simple, as well as R&D Systems, our sister brand, and Tocris, Novus, and the latest addition to the Bio-Techne family, Advanced Cell Diagnostics. Protein Simple is the brand of the Protein Platforms division of Bio-Techne, one of a whole host of brands that's applied across the entire life science field. This gives us a global reach with a very high install base of instruments around the world and employees and facilities around the world that can support you in your research with local support and local sales. In this simplified view of the life science world, you see our view of the life science world goes all the way from basic research out to diagnostics as well as pharmaceutical production. Whereas Bio-Techne doesn't actually perform this basic research, do the diagnostics or the pharmaceutical production. We support all aspects of that life science world with instruments, reagents, and consumables to make that work easier and more effective. One of the primary tools used throughout the world in performing research is the Western blot. You see a Western blot here. And we'd say about 1 in 2 life science researchers around the world do Western blots on a regular basis. This means there's up to 450,000 of you around the world who are doing this research all the time. We think that Simple Western is the future. It's the answer to this scientific question, the scientific process, because it allows you to simply prepare your samples, click Start, and get your results automatically. Highly sensitive data in a fully automated way that's fast, allowing you to perform an experiment in only 3 hours. The results are consistent, allowing you to do full quantitation, and it can give you that answer from only 3 microliters of sample. In order to perform this, we use capillaries instead of blots. What we do is we introduce a sample into a capillary. We separate that sample, either based on the size of the protein to be analyzed or its charge. We then capture the proteins after they've been separated to the wall of the capillary by exposing it to UV light and capturing the proteins onto a proprietary internal coating on the wall of the capillary. And then we perform a standard immunoassay. And then finally, we quantitate that signal using a chemiluminescent readout. There are 4 systems in the Simple Western product family. Wes, which can do a size-based separation on 25 samples at a time. The Sally Sue system, which can do 96 samples at a time in a size-based separation. Peggy Sue, which can do either size or charge on 96 samples. And then finally, the NanoPro 1000, which can do 96 samples in a charge-based separation mode. All of these systems deliver sensitivity, which we would say is greater than that of a traditional Western blot. In this example, you see 4 times more sensitive detection of a PI3 kinase. Now, different systems will provide different sensitivities. Those sensitivities will vary based on the antibodies as well as the samples being analyzed. But in all cases, we would expect better sensitivity than your traditional Western. This combination of highly sensitive assays and a small sample volume allows you to get data from just a few cells. So a very small sample size will allow you to get a great amount of data. This data will be delivered with consistency, low CVs across a large number of samples. It also allows you to reproduce that data between different days, different users, and different facilities. Sister brands, Novus Biologics and R&D Systems, we validated over 1,800 antibodies on the system and offer these through those sister brands. With our Sue instruments, you can get advanced throughput, giving you the ability to generate 96 Westerns in 30 minutes of hands-on time. All of that with great CVs. And you can also attain additional productivity. Looking at only 3 to 5 microliters of sample in a single run, you can actually look at an entire pathway from that run. So you can get information on up to 8 different proteins in a single sample in a single day, which would take you weeks and much more sample in a traditional Western. We call this gel-free, blot-free, and hands-free. And we continue on an ongoing basis to improve these systems, introducing new software and new reagents to enable you to look at samples in new ways and get more information out of those precious samples that you've worked so hard to generate. I'm going to turn things over to Dr. Chris Hager, who's going to describe the latest improvements to the Simple Western system and give you some more details on how you can get these improvements in your lab.
Thank you, Bob, for that great introduction, and thank you all for attending today's webinar. So far, you've listened to Bob give an introduction as to who we are at Protein Simple, what tools we can offer you for protein analysis, and specifically a bit more detail about what the Simple Western platform can do for you. But we also listened to you, the research community, as to what you need from us. We know that our customers appreciate it when we listen to them, but more so if we can listen and understand their needs and then take action. We've always pushed ourselves to follow this ideal. That's how we make better products that are better suited to fit our customers' needs. And so today I'm here to tell you about some of the new things we've done. As we all know, the holiday season is right around the corner. It was quite the weekend for shopping. Driving around this weekend, I saw Christmas trees on the rooftops of cars and, of course, a lot of holiday shopping. People everywhere were shopping for gifts, both large and small. And the first thing that I'd like to tell you about is all about small sample sizes for Simple Western. Sample cost is a really important factor for any researcher. The fewer available cells, the more expensive they become. Stem cell researchers understand this all too well. Stem cells are one of the least abundant cell types, and consequently, they are challenging to analyze. We spoke with Dr. Reiger at the University of Frankfurt, who studies stem cells, for his perspective on analyzing them by Western blot analysis. In order to obtain murine stem cells, Dr. Reiger's lab starts with a mouse, of course. They isolate the bone marrow cells from the thigh of the mouse, and then through various stem cell enrichment techniques, isolate approximately one stem cell for every 7,000 bone marrow cells. It takes a lot of work and a lot of time to get these stem cells, and in the end, a single mouse yields only about 1,000 sorted bone marrow stem cells. Needless to say, these sorted cells are extremely precious. 1,000 cells isn't a lot for most protein analytical tools, especially something like a Western blot. According to Dr. Reiger, more than 50,000 stem cells can be required per well for a traditional Western. Since a mouse yields about 1,000 stem cells, that means that around 50 mice are needed for a single lane. That is really expensive, with the purchase cost of a research mouse being anywhere from $20 to $500. That means these stem cells cost anywhere from 2 to 50 cents each, without accounting for the labor and time spent sorting the cells, or for housing and taking care of the mice. Needing so many mice for a single lane makes it cost prohibitive to run a traditional Western for rare and often precious samples. Researchers have to turn to another method to analyze these cells, but what if separation by size of the proteins of interest is necessary? Well, that's where we can come in. As Bob showed earlier, Simple Western is more sensitive than a traditional Western and uses only 5 microliters per well. So less cells are needed for analysis. For Wes, that's a 10-fold reduction in the amount of stem cells needed. So that same 50,000 cells could be used for as much as 10 wells on Simple Western. The same could be said for either Sally Sue or Peggy Sue. If these higher throughput Sue instruments aspirate the sample one time, it's still 10x fewer cells than needed for a traditional Western. But Sally Sue and Peggy Sue can actually use the exact same 5 microliters of sample up to 8 times, further driving down the cost per well and thereby saving even more money. Even with the 5 microliter volumes, analyzing stem cells could still get expensive, and we want you to be able to do more with less of the sample. So we're changing our protocol. With the new protocol for Wes, there's a new lower sample volume requirement. Instead of 5 microliters per well, we now want you to use only 3 microliters per well. That's a 40% reduction in sample volume. The same great data can be easily achieved by this new lower volume and is illustrated here. We compared the normal 5 microliter per well to the new 3 microliter per well recommendation with our biotinylated ladder, which contains 6 proteins ranging from 12 kD to 230 kD. We ran 12 replicates on Wes to show the reproducibility using this new lower volume assay recommendation. Shown in the top left graph is the average peak area for each molecular weight standard using either 3 microliters, shown in orange, or 5 microliters, shown in blue. Both plated volumes give very similar data, and the overall CVs are both under 10%, well within the specifications of the Simple Western platform. If we now look back at how Wes compares to a traditional Western and consider the new assay protocol using only 3 microliters per well, that's a 14-fold saving on sample, and the cost of running that sample goes down even further. With this change, precious samples can be used for even more analyses. We are really excited to be able to empower our customers to have these precious, expensive samples run on Simple Western. I hope you are as well. The next topic I'd like to tell you about is how we've gone about solving a common chemiluminescence issue, signal burnout. Nobody likes signal burnout because it means you can't use the data for quantitation. Burnout occurs when there's sample overloading, substrate exhaustion, or a worn-out enzyme. Essentially, the only way to combat this on a traditional Western is to wrap the blot up and image it as fast as possible. But in reality, this blot needs to be redone and the assay needs to be re-optimized in some way. Then you'll need to spend another 2 or even 3 days getting a better semi-quantitative Western blot. And keep in mind that with film, the dynamic range is pretty small, so you don't have to be off by too much to be outside the linear detection window. We understand that these things happen is the virtue of using chemiluminescence. In reality, Simple Western users sometimes experience this too, since we are using chemiluminescence and our instruments are more sensitive than traditional Western. So we took action. Before I tell you what we did, here's another way to think about signal burnout. It's like the blinding lights in your rearview mirror when driving at night. To deal with it, you'd have to manually flip down the mirror to see less of the light. Newer cars these days have automatic dimming features built into the rearview mirror and can sense when things are getting too bright and then reduce the glare for you. And not everybody has this in their cars yet, but most of you have a camera built into your phone that handles this glare through something called HDR. So what is HDR? HDR stands for High Dynamic Range, and in photography, it works by first taking a series of images, each shot with a different exposure. HDR then automatically combines the best parts of the 3 overexposed, underexposed, and balanced shots to create the best picture. Basically, HDR is math at work. Now we've brought HDR to Simple Western. HDR is a new and improved default multi-image analysis option and uses information from multiple exposures to achieve good signal-to-noise while avoiding signal burnout. For our current customers, the older multi-image analysis algorithm is provided for backwards compatibility and direct comparison to older datasets, but the HDR method is the preferred method for all new analyses. Basically, we can do some pretty cool math too. Here's an example of how HDR expands the dynamic range on Sally Sue and Peggy Sue. Shown here are 2 sample concentrations for recombinant DNA K analyzed with HDR, shown in orange, or the multi-image analysis that currently exists, shown in blue. Since the 63.6 nanogram per mil DNA K sample is burning out, if you use the multi-image analysis tool, the resulting quantitation is very close between samples that are actually 10-fold apart in concentration. With the new HDR method, a truer picture of the sample dilution is seen. Wes really takes things to the next level due to his small size. In addition to the new HDR detection profile, the compact architecture inside Wes allows for shorter time between substrate aspiration from the plate to the time of image acquisition. Faster time to imaging means better data. And second, Wes now refreshes the luminol peroxide substrate mix before every exposure, so there's new substrate for each image. Let's take a look now at some example data to see how these changes can improve your Wes data. With Wes's existing multi-image analysis, a saturated DNA K titration looks a bit counterintuitive. Instead of an oversaturated signal, like on a traditional Western blot shown on the right, a lack of signal would be observed with higher amounts of sample. New HDR in Wes, signal saturation is easier to identify as it looks much more like the saturated traditional Western I just showed you. What this means is if you are already familiar with signal saturation on a traditional Western, our new HDR makes it more intuitive and easier to recognize on Simple Western. Let's take a look at that DNA K titration in our graph view. In the multi-image analysis view, when loading either 636 microgram per mil or 1,000-fold less recombinant DNA K, you see no signal due to signal burnout. Only when significantly less protein is loaded, in this example another 1,000-fold less DNA K, can a true picture and peak for DNA K be observed. With our new HDR detection profile, you will see overloading like you would on a traditional Western. From these bottom graphs, it becomes clear that 636 micrograms per mil is way too much protein. There's a number of extra peaks and poor resolution. When a 1,000-fold diluted sample was looked at using HDR, now we see a strong peak in the 636 nanogram per mil sample. And diluting the sample another 1,000-fold really looks like it's part of a dilution series. As I mentioned earlier, HDR increases the dynamic range for detection. For recombinant DNA K, the dynamic range is increased to 5 to 6 orders of magnitude. Comparing to the multi-image tool, where loading more protein actually can decrease your signal due to burnout, HDR keeps the signal up. This gives a more intuitive understanding of whether you've got too much sample on the plate. If you compare the low and the high end of the titration curve for multi-image analysis, they appear to have very similar signals, which really doesn't make sense. Now with HDR, more protein is more signal. Let's now look at what happens with an endogenous lysate for detection of a 14-3-3 protein. Using the original multi-image analysis, the HEAL lysate dilution series doesn't really look much like a dilution series at all. Consequently, the quantitation, especially with more protein loaded, is inaccurate. With a new HDR method, a more true dilution series can be observed, and the detection dynamic range increases by approximately 2 log orders. Here's another great example of endogenous target detection and how HDR increases your dynamic range. Shown here is a comparison using a lysate detected for GSK3 alpha on Wes with HDR versus a traditional Western. I think what's readily appreciable is that they look similar, even at significantly higher amounts of lysate than typically is loaded onto Wes. Of course, with Wes being more sensitive, you can detect 10-fold less protein than a traditional Western. So the dynamic range is now extended on both sides. Comparing the quantitation from the previous two images for GSK3 alpha, a significantly wider dynamic range is detected on Wes due to Wes's greater sensitivity and the use of the new HDR profile. This means better detection and quantitation over a larger sample concentration range. When selecting an HDR assay in our software Compass, the plate layout and template are exactly the same as before. But when you go to the detection profile of an HDR assay, you'll see a checked box for high dynamic range and fixed exposures that cannot be changed. This assay has no delay from substrate aspiration to image acquisition and pushes Wes to get the best data yet. Since Wes is also refreshing the substrate before each exposure, he'll need a little bit more of the substrate. Instead of 10 microliters, you'll need to add 15 microliters of substrate per well. So this is a slight change to your plate prep. But don't worry, Wes will remind you before starting an HDR assay to make sure there's sufficient substrate for the run. We will also be including more of the substrate in the kits. Once the HDR assay has completed, the data can be analyzed with either the HDR selection or an individual exposure from the HDR exposure series. So you still have the flexibility to use our HDR profile or an individual exposure to look at your data. It's important to note that the original detection profile can still be used and you can choose specific image exposures. It is important to note that the delay between substrate aspiration and imaging occurs just as it did in earlier versions of Compass. These so-called original runs can be analyzed with the pre-existing multi-image analysis tool, the new HDR method, or with individual exposures. So the big question is, how and when can you get these improvements? If you are still doing traditional Westerns, I think our new lower sample volume requirement and increased dynamic range are two great reasons to go and buy an instrument. If you buy a new instrument, these features will be included with your shipment. If you're already a Simple Western user and have an instrument, for Wes, install the new version of Compass 3.1 and upgrade your embedded software. If you have a SU, simply install Compass version 3.1. And when are these features available to you? Starting December 15th. We're really excited to help you achieve a wider dynamic range for your assay and use less sample on Simple Western. We've brought you some great new features, starting with our high dynamic range and our substrate refresh for Wes. And we've shown you how with Wes, you can decrease the sample size needed from 5 microliters to 3 microliters, which is a real cost savings for those of you with precious and often expensive samples.
Thank you, Bob and Chris, for a very interesting presentation. Let's now move on to the last portion of the webinar, the Q&A session.